Hello, and congratulations 2020 South Carolina Junior Scholars. Indisputably, what the world needs more of right now are scholars. So pat yourselves on the back for being beacons of light shining brightly on our future. Anytime I want to take a glimpse into the future, I look at today. Today, Richland School District 2 has some 130 eighth grade students who have met the qualifications to earn this highly prestigious designation. The purpose of the South Carolina Junior Scholars Program is to identify students just like you who have demonstrated that they possess unique abilities to excel to exemplary levels academically, and for school systems to provide opportunities that will facilitate their intellectual growth, broaden their individual interests, and promote their scholastic achievement. Your Richland 2 family prides itself on facilitating your intellectual growth by creating, sustaining, and investing in a culture and environment of excellence where each of you are afforded the opportunity to maximize your gifts and talents. As you continue to do your part by making the grade, being good citizens, and celebrating your accomplishments, we will continue to do our part to provide the best education possible for each of you. We will deliver the premier learning opportunities that help you succeed and follow your dreams. So as iron sharpens iron, so one person sharpens another. Scholars, please continue to sharpen each other and those around you. Our future is depending on it. Continue to make your parents and Richland 2 family proud. It is my honor at this time to introduce our guest speaker, Dr. Michael Wyatt, a professor at the University of South Carolina's College of Pharmacy. Dr. Wyatt holds a Bachelor of Science and Master's of Science in Chemistry from Furman University and a PhD in Molecular Pharmacology from University College London. We thank Dr. Wyatt for joining us and helping recognize our South Carolina Junior Scholars.
Congratulations to you, the 2020 Richland 2 Junior Scholars. You're approaching an exciting and maybe a little scary move from middle school to high school. You have distinguished yourselves with your academic achievements, and this means even better things are possible with your studies in high school. Of course, the current pandemic has changed all of our lives now, which may add some uncertainty to your step up to high school, but let me reassure you that we will be able to persevere and become even better people because of how you adapt to the current uncertainty. I'm also confident that science and technology can help us in many ways through this current crisis. I will talk about this a little. My name is Dr. Michael Wyatt. I'm a professor and researcher in the College of Pharmacy at the University of South Carolina. I'm in a department called Drug Discovery and Biomedical Sciences, and the topic of my talk today will talk some about biomedical sciences. It's the science of medicine and health. I was honored to be asked by the district to say a few words that will hopefully spark some curiosity in each of you. So how did I become a biomedical researcher? Well, I started off with a lot of science in high school. I had chemistry and biology and physics, and I also had a computer science class. And then from there, I went to college, and at university, I became more interested in chemistry. I loved looking at molecules. I loved thinking about molecules. The more chemistry I took, the more I loved it. And that is one little piece of advice I can give. Take lots of courses in high school as a way to find your passion, find what you find really interesting, and then do the same in college. And so as I took more chemistry and realized I liked chemistry, I liked it so much that I kept going. And I earned a master's in chemistry also. At this point, I became a little more focused. This is where, during my master's project, we were synthesizing molecules that we were hoping to be anti-cancer drugs that could kill cancer cells. And this is where my research went more closely into biomedical research, because then from there, I went and earned a PhD. I also had a very unique and special opportunity to travel overseas as part of this research collaboration at the University College London. And so I did my PhD in pharmacology. And for those of you who are wondering what pharmacology might be, pharmacology is the study of drugs or drug action. How do drugs work? How do they work in the body, in cells on a molecular, cellular, physiological level? And not just medical or biological effects, but also side effects too. Adverse effects can also be caused by drugs. So that's what pharmacology is. And then for my postdoctoral work, I studied toxicology. And as you might be able to guess, toxicology is the study of poisons. And of course, sometimes drugs can be poisons too. If they're taken incorrectly, if you take an overdose of the drug, that can be a problem. And so there's some interesting comparisons there, but we wanted to certainly understand what might make some molecules poisons and some molecules drugs. But my overall passion is for science. And so I became a professor in the College of Pharmacy here at the University of South Carolina, and I've been here ever since. I love learning. I cannot help it. I love learning. And I'm guessing you too love learning based on your academic achievements so far. So learning about something that's never been done before is what I'm most interested in science. I love observing, learning, modeling, testing, and then reproducing results of others and our own experiments that we do in the lab and what people do in other labs to learn more about how things work and how we can maybe help people improve lives and improve health through biomedical research. Now, engineering is applying science, and sometimes they're talked about differently, but there's certainly a lot of overlaps between engineering and science because there's a lot of the same methodologies and thought processes that are very important. So learning, modeling, testing, and scaling. And by scaling, what I mean by that is thinking about different scales of how things are done. Usually in research labs, we're doing very small scale reactions. And sometimes engineers have to think in much bigger scales too, go to bigger scales. And of course, it doesn't always have to be start small and go big. Sometimes things start big and go small. And so let me ask you a silly question here for a second. I'm gonna stop this and ask, does anyone know what this is? Have you ever seen one of these? This is called a telephone. They have wires, and they used to have wires that would connect them to walls. And you think about that as a technological development. And then at some point, telephones went to these looking things. These used to be called flip phones, right? And so you think about the technological development that happened there. Pretty amazing technology, right? Let me go back to sharing my screen here. So, right, so technology engineering, okay? And so there was a famous person who came up with this idea of what's called Moore's Law. And a name for the person who came up with this idea, but the observation across many years of technological developments in the area of computer science. So if you look on this chart here, you see on the x-axis is years, number of years from 1950 all the way up to estimating up to 2020, where we are right now. And look on the y-axis here, get an appreciation of what's on the y-axis. Look, it's a log scale, right? So it's power of 10 on the log scale, from 1 all the way up to a trillion there and 10 trillion. And so believe it or not, computer technology began with machines the size of entire rooms and your entire classroom, maybe that you had in middle school. That would be the size of one of those computing machines. And technology over the years rapidly shrunk the computing power into smaller and smaller devices. And so that's where we went to computers that would take up an entire desk, called desktop computers. And then we went to laptop computers. And now we have, you can look at your smartphone that you or your parents have and see the computing power in that device is way beyond what used to take up entire rooms. And so there's been an amazing technological development here. And so technology impacts our lives in so many different ways. And this has been a tremendous development just over the last 50 to 70 years here, what's happened with computing power. Yeah, I said I'm a pharmacologist by training, and I've always thought about chemistry, but the chemical molecule I've loved the most is this molecule right here called DNA, or deoxyribonucleic acid. It's an amazing molecule. It's a beautiful chemical structure. It's also immensely powerful because it's the storehouse of our information. It's where our biological information is stored in every single cell in our body that stores information. And you look at the structure, and what's also amazing about it, it's only a 4-letter alphabet, right? It's only 4 letters in this chemical alphabet that stores the information. And it's an amazing molecule. I've studied it pretty much my whole career, all the way back to my bachelor's in chemistry, because we were studying molecules that would bind to DNA. And DNA itself is organized, in cells, in human cells, on chromosomes. So I'm sure you've heard about chromosomes before. And you inherit one chromosome from one set of chromosomes from each parent. And our DNA is organized, and it's also tremendously large in terms of what you're looking at there, because this is 3.3 billion bases and times 2 chromosomes for each chromosome. And that's over 6.6 billion pieces of information. So when I started college, this seemed like an insurmountable challenge, humongous information that would take forever to process. And it was a huge, immense scientific effort to sequence the human genome. When I started college, it seemed like going further than to the moon and back. But what was amazing about this process is that, believe it or not, starting in the mid-2000s, the cost of DNA sequencing actually beat Moore's law. Remember the law that talked about going from computers the size of rooms down to computers just in your smartphone? Well, the technological developments in sequencing DNA has gone even faster over the last 15 years. It's been an absolutely a stunning development. So again, look at the axis. I'm always a big fan of looking at what's on the axis of any charter plot. And we have years again on the x-axis there, just like we did in the other plot. But on this time, we have the cost of sequencing. And look, it's also on a log scale. So from $1,000 all the way up to 100 million. So that first human genome that was sequenced cost a couple of $2 billion for all of science to sequence it, and the costs have come down incredibly rapidly ever since then. And so this dramatic cost in sequencing has been an amazing technological development. And there's so many different technological developments I don't have time to talk about that have been good things coming out of this advancement for medicine and treating disease. It's been a really fun development to have lived through as a scientist during this process. So, okay, let's go back a little bit in time just to a different technological development that helped that advancement in cost of sequencing. And it's called the polymerase chain reaction, or PCR. And it's a way of amplifying DNA, making more DNA, and it works through an amazing way of being able to exponentially amplify DNA. So remember, we talked about log scales there. We're talking about exponentially amplifying DNA, where each product of this reaction becomes a template for the next reaction. So for each number of cycles you do, you're exponentially amplifying DNA. So from 16 to 32. You may have heard the story of the king's chessboard and putting one piece of rice and then doubling that on each subsequent box of a chessboard, and by the time you get to the 64th box, you have a tremendous amount of rice kernels, way more than you can fit onto a chessboard. And so it's the same application here. And so typically in the lab, we are only going up to 30 exponential amplification cycles, but that still gives you over 1 billion copies off of one single piece of DNA template. And so how that's being used right here, right now, is with one extra first step, we can also detect RNA too. So why am I bringing up RNA? Well, the coronavirus is an RNA virus. And so we can use science and technology to detect it, to be able to help people. And so we can take advantage again of technology, not just scientific discoveries in the lab, but technological advances. And so we can use different machines in the lab. And what I show here on top is a 96-well plate. It's a plate that can hold 96 different samples, and we use these a lot in the lab. In fact, we also have plates in the lab that can hold 384 wells, 384 different samples in a well, and do lots of analysis simultaneously. That's a tremendous development. And this is where also engineering becomes very important too, because now there's also technological advances in engineering that create robots, machines that can process stacks and stacks of these plates holding 96 or 384 samples per well, and a tremendous amount of engineering developed to be able to more rapidly advance testing. And so we now get to the point where how can we put all this together to help society more broadly? And this is what's so exciting about what's going on right now. Despite the seriousness of the pandemic, we're very optimistic we can begin testing and figuring our way out of this pandemic. And so one example would be a PCR-based testing strategy, where you're collecting samples and isolating RNA, amplifying it, detecting it, and then reporting and letting someone know if they have the virus, and then being able to provide medical treatment, to be able to provide isolation so they're not spreading the disease. But this whole thing requires so much more than just science. It's also the technology, the engineering, and there's much more that goes on here too. We need doctors, pharmacists, nurses in the hospitals, working in a healthcare setting. We need public health officials. We're watching what's going on at a population, amongst the whole population. We also need people who are called bioinformatics experts, people who can use computers to examine tremendous amounts of data. Bioinformatics is an area of expertise that there's going to be a huge need for, as we're in this data era where so much data can be generated with these new fancy techniques. We have to be able to not just generate the data, but be able to analyze it and process it and make sense out of it, and then plan new things from it. So analyzing data is very, very important too. And so many exciting opportunities in science, medicine, and technology. I hope whatever simple advice I could give, I definitely encourage you to find something you find so fascinating and interesting that you can't help yourself. You want to learn more about it. Finding something you are passionate about goes a long way to finding success in life, being able to find something that you find so interesting you can't help it. The other thing I personally enjoy about my career is working with students at all levels, from high school to undergraduate to graduate students. And so this is just a picture of a few of the people who have worked in my research lab over the years, doing all sorts of fun and interesting research. And I do need to acknowledge my mentors from my undergraduate and graduate and postdoctoral research projects. Some people very important in my life. That's another piece of advice I can give. Always look for good mentors, people who are willing to give you good advice and help you. And so when you're in high school and taking classes, don't be afraid to talk to your teachers and ask them questions. It's so important to be able to be unafraid to ask questions. And so with that, I'd like to go ahead and once again congratulate you, the 2020 class of junior scholars, and best wishes going forward into the new academic year.
Shahil Agar Wall, Alyssa Amaker, Lillian Amick, Jia Anand, Harsha Avula, Marguerite Bagting, Jordan Bamin, Mehul Belakati, Jordan Blanton, Kyra Booker, Ella Boston, Lane Broderick, Benjamin Brown, Rachel Brown, Sean Buzzhart, Aria Caldwell, Zanaya Castile, Peyton Cermak, Sharif Chada, Everlast Chagoba, Cameron Klein, Bryce Cruz, Rylan Cunningham, Lindsay Doherty, Mary Jane Davis, Mithim Deva Rapali, Sanvi Devakar, Jackson Dowie, Abigail Dubberly, Garrick Edward, Taylor Edwards, Elise Fields, Zachary Freeman, Xavier Galloway, Shrihan Ganesh Babu, Alexa George, Paris Grant, Anna Graves, Abigail Greer, Jace Griffin, Amir Guess, Brant Hagler, April Harbison, Kaylee Haberlin, Bryce Hermesh, Micah Hicks, Emma Hook, William Horman, Elizabeth Huffman, Yojit Jundul, Rachel Yochin, Elizabeth Johnson, Savannah Jones, John Kim, Jareya Kumar, Abby Lee, Jisoo Lee, Morris Lee, Alexis LaRuzic, Wilton Lever, Melissa Lauterbaugh, Abigail Lucas, Annabelle Manville, Blue Martin, Ariana McCall, Jacob McDonald, Megan McGriff, Chase McKenzie, Catherine Milburn, Ryan Miro, Savannah Mistars, Elijah Moskowitz, Bargavi Munagala, Ellis Murray, Sophia Myrick, Jamila Kanate, Satvik Nilakuditi, Nathan Wen, Katie Norris, Katrina O'Grady, Evan O'Mara, Michael Papaletsos, Shivani Patel, Josiah Peoples, Eva Pendleton, Ava Phelps, Cade Polakowski, Madison Powell, Donald Proussel III, Patrick Quirk, Arvon Ramoso, Anna Rosolov, Joseph Ravindran, Corinna Rice, Reagan Richardson, Kristen Robertson, Cooper Rom, Aaron Rupp, Hannah Schaefer, Hunter Shishis, Journey Simmons, Marina Slawbog, Jason Sobel, Anna Sparrow, Sydney Sutherland, Lydia Tasso, Irene Tain, Brooklyn Taylor, Emily Taylor, Ivory Thigpen, Micah Tunnell, Samvit Urs, Kartik Valerie, Adam Van, Ashton Walker, Bella Ward, Ryan Ward, James Washington III, Harrison Weaver, Trip Whitaker, Yasmin White, Madison Whitfield, Marguerite Whitley, LaDeja Williams, Jordan Worthy, Kendall Wright, Sashwat Yadav, Drayden Young, Ryan Young, Lily Zhang,